So welcome everyone to this exciting panel on the life of a patient sample, from donation to discovery. So again, I'm Joan Levy, I'm Chief Science Officer of MRA, and it's my pleasure to moderate this panel. So I'm just going to give you some background that'll lead into our biorepository and biorepositories in general, what they're used for, how they contribute to research, and after this intro, I'll join the panel. Okay, we're going. So the MRA is the largest private funder of melanoma research worldwide. We've supported about 175 million. We've invested that in over 500 different research projects, and we have supported research in over 17 countries. Why am I telling you this? Just to give you a little bit of background, but the way I want to craft this panel is to say that a significant proportion of our research investment is really dependent on the availability of donated patient samples to either be directly analyzed or to be used to create these preclinical models that we could do a lot of different types of testing. So what are our MRA's interests? We have three major strategic research areas. One in the rare melanoma subtypes of acral, mucosal, and uveal melanomas. We have significant strategic investments in these rare melanoma subtypes. About 14% of what we've invested is towards research for rare melanoma subtypes. We also have a rare melanoma registry, which is a direct-to-patient registry for acral, mucosal, and cutaneous melanoma patients. It's been open for about 3 years, and many of the patient advocates and scientific and medical advocates, and I should say advisors, are actually in this audience today, and we, on behalf of the MRA, we want to thank them for their support, because we have now about 670 participants in our rare registry. And this is to collect patient-reported data right now, as well as clinically abstracted data, and we're also going to connect them to the new MRA biorepository. Our second strategic investment is based on the fact that the melanoma field has done incredible, 17 new drug approvals since 2007. That's fantastic. But on the flip side, we have to say that about 50% of those melanoma patients, unfortunately, will either not respond to their therapies or progress during treatment. So therefore, we still have a lot of work to do to help those 50% of patients not responding. And finally, we have a focus on using AI and other types of machine learning that you've heard about before to improve early detection and diagnosis. So I'm pleased to say that MRA not only funds research, we actually are actively engaged into research. We, again, I just described the rare registry. It is supported by a Department of Defense grant we were fortunate enough to receive. We also have another grant through the DoD on studying risk factors for rare melanoma subtypes. And recent publication from this particular grant demonstrated that for acral melanoma, there's a 30% increased risk of developing acral melanoma if you were exposed to Agent Orange. So that just came out in publication, and our CEO, Mark Hurlburt, as well as collaborators at the VA, as well as Mass General, have been involved in that project. And finally, we have support to open what I'm now going to focus on is our MRA biorepository that has just opened last November. So many of you might want to say, well, what is a biorepository, and who has these biorepositories? I'm sure most of you know about them. They are really places that store clinical tumor samples and normal tissues from blood, from cheek swabs, and they store them with associated biological and clinical information and are housed at different facilities. And when we talk about, and what we'll talk about today is donated tumor tissue, what we really are referring to is tissue that is no longer, residual tissue that's not being used for diagnostic purposes, or that the patient is not using for other types of genomic analysis. So it's really residual tissue that's just being stored that, as you'll see, patients consent to be used for research purposes. And this just depicts a little life cycle of a patient sample. It starts with a patient consent. This is voluntary. It's so patients consent to donate their tissue samples. They are taken from different medical and surgical procedures. Dr. Beasley will talk about that. And then they're acquired at different facilities, stored, processed, and then they're released for different types of research studies. And, you know, the key principles here is that it's voluntary participation, no cost to patients. There's privacy protection. When they're released to researchers, they're de-identified, so researchers can backtrack and know who the sample was from. And it's really, you know, for patients to help contribute to research. So our MRA Biorepository was launched last November, and this is to help support research by donating tissue samples. And our biorepository is being housed at the University of Colorado. They have incredible expertise in collecting melanoma samples. They've collected over 5,000, and they were perfect collaborators to help us with this initiative. And it is for patients with all subtypes, even though our focus is going to be more on patients with acral, mucosal, and cutaneous melanoma, because there is another biorepository that's going direct to patients to collect samples for uveal and ocular melanoma. It's really 4 steps, and the participant is only involved in the first step, which is if you're interested in donating your tissue samples, you can just email the email shown above. We have postcards and all about this and a landing page, and the MRA will send the person, interested party, a consent, and if the patient is interested in participating, they sign the consent, return it to us. We send them a form so we can get information on where the sample is, who the doctors were, who the hospital is, and some clinical information. And then basically, step 1 is over for the participant. We take it from there. We actually use that information to contact the hospitals and doctors to get the samples and have them collected and sent to the University of Colorado. Once received by the University of Colorado, they then are stored, processed, and ultimately the main purpose of collecting these samples, especially for rare subtypes, is so that researchers can submit to us different research studies that they want to use these samples for. And we have a tissue oversight committee that will review those research proposals, see if they're, you know, important research for the use of samples, and they're qualified researchers to do the studies, and then we will release the samples to these research studies. So basically, for participants, just step 1, sign a consent, send in a form, and then we take it from there. So we're advertising this. Again, we have postcards. We have, it's advertised, I'm sure you see it in our e-newsletters. Rare registry participants can donate their tissues, and we will link the information from any tissue information to their participant entered information, survey information, as well as any clinically abstracted information. But it's not only open for rare registry participants, it's open to everyone. And Renee is doing a fantastic job in advertising this through social media, and we have a website too. And I just want to say, I mentioned that there is another biorepository, and that's for uveal ocular melanoma. It's being run by a Cure site. And so for any uveal patients, you have the option to donate tissue to their biorepository, and pattern.org is running this, and pattern.org is part of the Rare Cancer Research Foundation. So at this point, I am gonna stop, and I am going to join our panelists, who I'll introduce briefly and then ask them for an introduction. So sitting here is Dr. Patricia Possik from the Brazilian National Cancer Institute, and she'll briefly introduce in a minute what she does, and Dr. Georgia Beasley on screen, who is from Duke University, a surgical oncologist who really acquires samples and uses them for her research studies and for other research studies at Duke. So I'm going to sit down right now. Okay, so Patricia, would you like to give yourself a better introduction than I did, and tell us what you do?
Thanks so much, Joan. Well, very happy to be here. I first want to congratulate the MRA for this great initiative of creating this biorepository. I think it's going to have a huge impact in the field, and I'm very thankful for that. I'm Patricia. I work at the Brazilian National Cancer Institute. I'm also an international fellow at the Sanger Institute in the UK. And since I've started my lab in Brazil, I focus on acral melanoma. For the past 8 years, we've been collecting samples, recruiting patients, collecting samples, and then we are characterizing both the patient sample and also models that we develop from these samples. So we put we turn these samples into cells and into tumors, and then we study them, and we also compare with the patient sample.
Okay, thanks so much. Georgia.
Hi, Joan. Hi, everyone. I'm sorry that I cannot be there in person. Also express my excitement and appreciation to the MRA for starting this wonderful initiative. So I'm a surgeon, so I'm often the one that literally does the collection of various samples, and then I also work with scientists, so I kind of get to be on a couple different places during the life cycle of the patient sample. And hopefully today we can talk about, some of the common questions that patients have when they're donating, about tissue that remains for what, future things and all that. So I think there's always some questions about that. So look forward to discussing all that with you all, and thanks for having me.
Yeah, so just for everyone to know, almost every large academic institution has their own biorepositories. And, and now foundations are starting to go direct to patients to create their own biorepositories to collect, as I mentioned, donated tissue to be used for research studies. So I'm going to start with you, Georgia, and say, to better understand patient donations. Like, a patient comes in, you're a surgeon, I'm sure they have to sign consent for surgery, for having samples sent to the pathology lab. So at what point do you ask a patient, at what point do you talk to a patient about donating their sample for research purposes, separate from what they're doing?
Yeah, I think to be honest, it's sometimes very hard because I know patients, they're incredibly courageous, and oftentimes, they're facing a rough diagnosis, especially if I'm meeting them as a surgeon, usually they have, more advanced type of melanoma. And so obviously they're already a little bit nervous, and we're talking a lot about the surgeries and the treatments. So sometimes it is difficult to bring it up, like, oh, by the way. But I think most patients are really interested in at least hearing about it. And I think from my standpoint, I try to also sometimes do it a little bit later if I'm feeling like someone's really overwhelmed. There are some times it's just not the right time, right? So I really do consider that when I'm first meeting the patient. If they're feeling pretty overwhelmed, sometimes I'll give them a little bit of time. If we're getting to a good place, I'll often introduce the concept of donating tissue or blood. And I think one of the most important things for me is that patient trust that we can never just take for research when they need it for whatever clinical diagnostics. So just to get very detailed about it, if somebody has something the size of a tennis ball, I cannot pass off the whole tennis ball for research. Almost 90% of it really needs to go to the clinical lab so that they can run all, like I was listening to the last panel, they need to be able to have it for all the sequencing, and they need to have it for anything that gets discovered. So one of the most important things that patients should know when they're signing up for the institution or whatever, and they should ask, is that the tissue should never be used up by research. So I kind of promise, patients that, and to be honest, I would say, again, back to the tennis ball size, I'm probably taking 10% for research. The rest, I feel strongly that the patient should have, and it should be stored in case, in 5 years from now when we need to test the next best thing, that it's still there. So it can never be used up, but I would encourage patients to ask about that. A lot of patients ask about the benefits, and I'm very honest about that. You can imagine if you're collecting patients, 500 patients, it might be very difficult to kind of get that research result back. So I do sort of tell patients when they're donating, they're likely doing it in an altruistic way, meaning it's probably unlikely to improve their own treatments, but they're definitely going to be helping, a lot of other people. In rare cases, it might, but again, just in full honesty and disclosure, I often tell patients that they are, but they're making a major contribution to the treatments. You think about all the things that have been developed. I can name so many. All the gene testing that we've done, it was all done on tumor samples, circulating tumor DNA. All those things required patients being able to have that altruistic and donating their tumor. So we take it very seriously, both me as a surgeon and all the researchers that I work with. We certainly understand the sacrifice that the patients are making, and we greatly appreciate them stepping up when it's a very hard time.
Yeah, thank you. Patricia, I know you're in, the research part of it, but in Brazil, I mean, do you know how clinicians approach the patients, and if there's eagerness to do it and to donate samples?
I think it's, it's universally basically the same process. Depending on the institution, if they have their own biobank, they have a whole logistics ready to help with every step. So we have, apart from the surgeon and the pathologist who's going to decide what is this 10%, you also have a nurse or someone who assists with explaining to the patient why we want to collect the sample and how we're going to store and what type of projects will use this sample. And then if the patient consents, then we get a okay to collect the sample and to press play on these logistics. So it's very similar. The difference is, like for example, in our case, that we want to collect different types of samples. From this 10%, we want to collect for the genetic analysis, but also part of this piece, we want to keep it alive so we can bring it to the lab and generate models. Then we need to also have a separate infrastructure for that, because the way you collect the sample is different. So I think that's the only thing that I would add.
Yeah, so Georgia, what are some of the misconceptions that you hear from patients? What are their fears of, are there fears in terms of donating the samples or
Yeah, I think
lack of understanding?
Yeah, I think, I sort of addressed a little bit about they want to be sure that, and again, because like you mentioned, the advancements are happening so fast that, what if a new discovery is made and they don't have, their tumor it's been used up, right? And so that is, again, something that should be a never event in my book. The other thing, and if you don't know the story, it's pretty interesting, related to HeLa cells. And so this patient, those happened to be the patient initials, so there were certainly no privacy rules back then. So privacy can be a concern. And I think we've taken a lot of measures to ensure that the samples are de-identified. And so I think patients have maybe a little concern about that, but I think we really try our best to make sure that that's not the case. And then, it always does come up again, if you don't know the story of the HeLa cells, but basically the researchers took cells from a woman and they're still used for science today and have made millions of dollars, selling them for research use. So I think patients do sometimes just wonder about, how that all would work if that were to happen, and I don't have a great answer for it. We haven't, quite sorted out that if your cells happen to lead to discovery of something. Unfortunately, I think, again, since we de-identify and all that in this day and age, I don't think that could happen. But I think there is, rightfully so, given some of those stories, a little bit of distrust about what's happening to the sample or whatever. And so I'm always, certainly telling people if they're uncomfortable, then that's okay. Like, we respect their decision, but also that the impact that they make has literally revolutionized, the field. And you mentioned the 17 drugs, got approved and all these therapies because people gave their tumor. You can kill a lot of cells in a dish in a mouse, but you have to have the human tumor to really make these discoveries.
Mm-hmm. That's a great point. And, before we go on to more about what the discoveries are, what the research is, I mean, sometimes the excess tissue is stored, the pathology stores it, and sometimes, as Patricia said, you need fresh tissue. So to point out, in terms of the biorepositories we're running and a Current site, we could collect archived tissue samples from patients who've had surgeries, and also patient samples that are undergoing future surgeries. So both types. But, how long, could these tissues be stored? And I guess for models, you need to get them right away. And maybe, George, in that answer, you could say, in terms of how long they could be stored, what can, say, archive tissue samples, like tissue samples that are in these blocks that the pathologist has sitting on their benches, what could they be used to do?
Well, and that's a tough answer because it depends on the test that you're running. And so, like you mentioned, a lot of the tissue is stored in paraffin blocks. And so we do know, just based on some of the work that we're doing, if we try to go back and do, I guess I'll call it more fancier things on the paraffin, like isolating RNA or whatever, there's definitely poor quality the older the sample is, right? So if they're 10 years or something. So the more aged it is, some of the more advanced techniques definitely have some issues regarding quality, which isn't surprising. But some of the more basic things, like, again, if you wanted to go back before the BRAF, sort of era, from 20 years ago, if you cut a piece of paraffin and just stained it for BRAF, it would still probably be okay, because that's a simpler thing. And so that's, again, why it's really important that we always have some of that tumor stored, because we might be able to go back and do some simple things for a number of years. I mean, paraffin is supposed to keep it stored for a really long time. But I will say, the fancier that thing that we're trying to do on the paraffin, there is, an age, factor in terms of degradation. So I hope that answered your question, because it does depend. But we can still do a lot of things with samples that are 10, 20 years old. We can do a lot of that are in paraffin. We can do a lot of basic things. But some of the more advanced things, maybe not, but there's still plenty to be done, even if your sample is a little bit older, stored in the paraffin.
Yeah, so I just want to move as we come to a close to really have people understand, and you've mentioned it, Georgia, what kind of discoveries we could make. So I'm going to ask Patricia, what can you do with these models that you can create out of fresh tissue? What sorts of discoveries have been resulted from those models?
First, actually, I want to just complement what Georgia said in terms of how long it lasts, because it's interesting that this 10%, if you take like half of it and then you make a model, it will last forever, because in theory these cells are immortal. So if you give them the right conditions, the right nutrients, or if you put them in mice, so that the mouse structure or environment helps it grow, you can then freeze it down in a way that it preserves viability, and you can use it for different projects, different experiments, basically forever. So that's even like a very small piece can be immortal. What was the question again?
So the question was, what kind of discoveries can you use models for? What's come out? Why do we create these models?
So what we can do when we have all of this information combined is you have the model, but you also have all of the information from the patient and the genetics of the sample. So once you know all that, you can ask questions about what makes this cell proliferate so much, or what makes it metastasize to certain organs. And by using the genetic information that you have from the patient sample, direct patient sample, you can modulate it either genetically or using agents that target specific proteins. You can study the process of how cells metastasize from when you mimic where the primary tumor is, and then you wait in a mouse, you inoculate in a mouse, and you wait and you see where these cells is going, why is it getting there, not into another organ. So you can study all of this with these models.
Yeah, and a lot of drug testing, has to be done in these preclinical models before that they could enter human clinical trials. So they're extremely critical and to test different drugs that are looking promising, and if they pass the check and look promising in the preclinical models, then packages could be put together to then go to the FDA to have them tested in humans. And we're doing all sorts of really cool models now, not just implanting on plastic or growing tumor tissue in mouse models. We also can form these organoid models, so they're outside of the mouse model. You grow them, and it's the tumor cells, but they also take other cells from the patient to make like a tumor microenvironment, so that they can see interactions of tumor cells with the cells that are surrounding the tumor and kind of mimic what's going on in human tumors. So, Georgia, what discoveries are you excited about? I know we talked about BRAF, but you've been involved in a number of projects that are really exciting.
I was just thinking here, like making my list. So some people may be aware, but a lot of the information we get from tumors, now that we can do this really advanced sequencing and pick up all the different mutations and then looking across populations, right? And so some of you may be aware, just this week, the NCCN guidelines added a Merlin like diagnostic test into melanoma. There's also another company's Castle, and Castle also has a test for uveal, right? Where now patients, in addition to the standard pathology, a pathologist gives you Breslow depth, et cetera, but now we have tests that add the genetics in. So that's all done on tumor. So it gives people more information about how likely their tumor is to metastasize or how likely it's spread to lymph nodes. And it really starts to help patients, just like the uveal test, can really guide patients' management. And that's all from tumor. So these are tests that involve getting a piece of tumor, and they've all been developed from patients' tumors. And they've now been able to help patients, guide management. So some people with uveal may be aware of the class 1 or class 2, and now in cutaneous melanoma, we have companies and NCCN now approved another test on your tumor to help figure out what your treatment's going to be. The vaccines, the melanoma vaccines, the mRNA vaccines, right? Those all are developed from patient tumors. The idea is you figure out what are the antigens or the mutations in the tumor. You have to know that before you can generate a vaccine, because it's specific to that very patient's tumor. And then finally, something that is also really exciting that most people might be hearing about is the idea of circulating tumor DNA. And so that's where a lot of patients now know that we basically start a treatment, and then sometime later we get these scans. And sometimes on these scans, it's really actually pretty hard to tell what's going on. Is the treatment working? Is it not working? It can be kind of hard. But the circulating tumor DNA is a blood test, and the blood test can follow whether or not the melanoma is still kind of floating around or not, and that all has to be developed. The only way they can detect it in your blood is they have to know what's the mutations and stuff are in the tumor itself. So that was all developed from patients' tumors, and in fact, to get a circulating tumor DNA, they have to get a piece of your tumor to generate, to figure out what it is, so they can test your blood for it. So those are 3 really great, I think, examples. Developing blood tests, developing more genetic tests. The vaccines, those all were done just from patient tumors, nothing else. And in fact, they need patient tumors to be able to build the test itself. So without people sort of, there always has to be a first. So without people kind of taking that leap and donating their tumor to get the test developed, we wouldn't have them.
Yeah, so basically all these tests that now are being used or approved originated from research on donated patient tissue samples that help lead to the to the ultimate development and use of the test now clinically. So that's a really important factor. So what I think we're going to conclude, I'm going to ask each one of you, and this I think was a great discussion, and unfortunately Dr. Beasley is not here with us, but I'm sure Dr. Possik will speak to patients who are interested in what she does. Is what should patients, if you had to sum it up in 2 sentences, what should every patient know about tissue donation and its importance? So I'll start with Patricia.
I think that it won't compromise the diagnosis or anything and related to treatment and to care. And when you think of benefits, the impact that can have on improving treatment for future patients is incredible. So I think that's very encouraging.
Thank you. Georgia?
Yeah, if done well and right, what the MRA is doing, it really should not jeopardize your care in any way. So that is always a priority for us. So we never can do collect samples, or if it's going to jeopardize your tissue. So in fact, just to give an example, if I'm taking something out and I really just it's too small, then I just say, that's it, we're not gonna send it, for research or something like that. So I think it can never jeopardize clinical care. And I'll just echo my co-panelists that the benefit for advancing, care for other patients is really limitless.
Yeah, well, thank you for those thoughts. And I'll just say, just to add to it, all the things we know about genomics, like what tumor biomarkers or other types of alterations in tumors, and now also in the cells in the microenvironment, came from research on donated tissues. So, and MRA has supported a lot of the genomic work across different subtypes, but again, those were from donated patient samples that were in there, different academic biorepositories. So I'm just gonna now thank the two panelists, but also open it for a few questions.
I'll bring the mic up. Hi, my name's Paloma. I'm a 3-time stage 4 acral melanoma survivor and EPO-NIVI trial. The piece that I'm concerned about, because it started growing again after 7 years, is in my liver, and I have my biopsy next week, I just found out. How can I ask them to give you a piece of it when they biopsy it? Or is that too small? Can I ask them to take a bigger piece?
Well, I think that's, well, I'll let George. George is a surgeon, I'm not. Yeah. I'm the nerdy researcher originally, so. Well,
first, very courageous of you, and always, just appreciate that, you're sitting here, what you're dealing with, but that you're willing and open to even listen and think about it is pretty awesome. So thank you for thinking about it. So it's definitely something you should discuss. I mean, Joan, this sounds like, one thing you do is ask the MRA, and you can sign up to give the tissue, and then ultimately, you just can chat with the providers, because in some cases, it might be extra risky. So sometimes when you're biopsying, the liver or whatever, it can be a little bit more risky to take an extra or more. And so if they don't think it is, sometimes it's not. And so if they think it's not going to, cause any extra injury or whatever, then that's fine. And there are some cases where it's really like a game time, sorry to use that, but, decision, where if it's all going well and they feel like they can take a little extra, then that's great. And usually it's pretty safe, I would say. But, then there are times when I'm sure they'll understand too, if there was any concern, for example, that they don't have enough to make the tissue diagnosis, then it can't happen. So a lot of times in those cases, what we do is we have the patient sign consent and just sort of be ready. And then while we're doing it, if there's extra, that's great. But if there's concerns over the clinical need or safety, then of course, we just don't do it. And that's perfectly fine.
Yeah.
But we definitely kind of get ready for it. Mm-hmm.
Yeah, so what you can do is there are postcards, you could contact us, and we'll send you the informed consent that you could read through, and then if you want to participate, you could then send us the information as to where the surgery is going to be done, and then it'll be up to your surgeon to see if additional tissue could be sent to us, and I'd be happy to talk to you. Any other questions? Sorry.
Hi, I'm Brittany from Pennsylvania. I'm also a melanoma survivor. And my question was similar to hers, because I understand and actually have confidence of sending you the patient sample and that you'll do what you need to do. I do have also more questions about the process of how do you bring up the conversation to your doctor? Do you even tell your doctor, or do you get a biopsy and then just come home and send you an email and not tell the doctor? Also, you mentioned some of the institutions have their own biorepositories.
Correct.
So who decides if you get it or if they get it, and is that why we just email you and we're not a part of that conversation? You just do it in the background?
So it's a good question. I will say that every patient the sample is the patient's sample, and it's a decision that a patient could make as to where their sample could go, except obviously if it's needed for diagnostic purposes, then it goes to the institution. All you have to do is, if you want to donate to the MRA Biorepository, you can sign the consent, we'll send information. We could contact the doctor directly, or the surgeon, and make arrangements to ship the sample. Some patients want to, I've been involved in other biorepositories direct to patients. Some patients want to give their surgeons a little heads up that this is what they want to do. But if you don't want to discuss it with your doctor at first, we will reach out, and if there's any questions, we'll contact you. But generally, all you have to do is the first two steps. So, and again, I'll be around at the end, so I can talk to anyone who's interested.
Sorry, John. I just wanted to add that, because you asked when, if you do that before or after. The earlier you do that, the better, because then they can prepare to collect the sample and also decide what type of collection kits they can send your surgeons to be prepared at the time of surgery.
Yeah, if you're having surgery, it would be helpful to do it as soon as possible so we can get the kits to the hospitals and surgery units for collection. But I'm going to end there.
We have one last question. Hey, Joan, over here.
I had a question. With the samples, they're de-identified, so the person's name is not on there. Is that correct?
Correct.
Okay. How about, I'm a mucosal melanoma patient, and is the samples identified by the type of melanoma, or is it just thrown in as general melanoma? So, in other words, is the research done, like mucosal melanoma, is there research done for that specific melanoma rare type?
So yes, it is, and any clinical information, like if it's a mucosal melanoma, if it's a primary tumor, if it's metastasized to a specific organ, is included. That's de-identified information, but any identifying information like your name or where you live or your zip code, things like that, are not provided to researchers. We store, MRA, being the sponsor of this study, stores that information safely, but we do not release that information to researchers. Any type of information that could re-identify the patient that donated the tissue.
And then the research is done for this specific subtype of melanoma?
So researchers have requested, especially for the rare subtypes, they need, they're very precious samples, and they always need more samples. And a researcher who's doing mucosal research can request, I need 10 mucosal samples of this type, primary samples or metastatic samples, and they would propose a research study. It would have to undergo approval, and then we would release the samples. Again, we do give certain clinical information, but no clinical information that could re-identify that sample to a person.
Okay. And how old can the sample be? You said it can be very old.
It really is It depends, but even samples that are stored in pathology can be used, for many years afterwards for certain types of assays. But we're getting better with those assays to use more archive samples. So I think I need to stop at this point, but happy to answer questions.
I just wanted to add one thing, because he asked if it's going to be used to study mucosal melanoma only, but it's important to know that sometimes it's very important to compare the different melanoma subtypes, because sometimes a certain treatment is developed for one subtype, and we assume that it can that will be efficient for different genetic and histopathological subtypes, and it's not. So comparisons are also important.
Correct. There could be other subtypes that are involved in the research studies as to do these comparisons between the different subtypes. Thanks for adding that, Patricia. Anyway, thank you so much. I thank the panelists. Thank you, Georgia, for joining. Thank you, Patricia, and thanks to all of you.
Thank you all.